Okay, I think we'll kick it off now. Can you go to the next slide, Shanice? My name's Craig Hardner. I'm Principal Research Fellow in the Centre of Horticultural Science, and I'm also the Chair of the QAAFI Science Seminar Committee. I'm also Chair of the Australasian Plant Breeding Conference that we'll talk about in a moment. Can you go to the next slide, please, Shanice? So I'd like to begin by acknowledging the country on which we meet today. The University of Queensland acknowledges the traditional owners and their custodianship of the land in which we meet. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australian and global society. Thank you. So our presentations today are from 3 students that will be attending the 16th Australasian Plant Breeding Conference, which will be held on the Gold Coast next week. This has been a long time coming, and with many things during COVID. And as I mentioned, I'm the chair of the conference. We originally planned the conference for mid-2020, and after several postponements, we're ready to join our community on the Gold Coast next week. So would you go to the next slide? So just a bit of housekeeping, as always. So the seminar is scheduled from 12 to 1. Each of the students will present for about 15 minutes, which is their allotted slot for the conference, and we'll take questions immediately after the conference, after the talks. So there'll be essentially 3 20-minute sections. Can you please include your questions in the question and answer session, not in the chat that I did a couple of weeks ago, and not in the chat function, and then I'll facilitate those questions. So Shanice, can we go to the next slide? So this is Shanice's introduction slide, and I'm just trying to find, okay, there we go. Okay, so Shanice is a PhD student in QAAFI with Centre of Crop Science. Her PhD research is using new innovative tools to increase the reliability and productivity of mungbean for Australian and Myanmar growers. So Shanice, I'll let you take it from there. Thanks a lot.
Great, thank you so much. So as mentioned, my name is, sorry, I'll just make sure, cool, not muted. Great. So my name is Shanice, and today I'm fortunate enough to present to you all the talk that I will be giving at the APBC conference, which is titled UAV-Based Technologies to Reveal the Secrets of Mungbean Yield. Sorry. So to provide a bit of context, mungbean is an important cash crop that over the past 2 decades in Australia, the total production has steadily increased. And for those who aren't familiar with this crop, it's actually one of Australia's highest value grain crops. And this increase in production has the opportunity to rapidly expand with a substantially growing plant-based protein market, with currently the Australian plant-based protein sector having a consumer expenditure of $185 million, as well as $2.7 million in exports. And as you can see here, this will significantly increase by 2030. However, despite this increase and projected increase of this crop, the expansion of mungbean is limited due to its low yield production and variability, as mungbean is highly vulnerable to many abiotic and biotic stresses, as well as displaying a number of physiological constraints. And this is the result of many current commercial varieties having a narrow genetic base, with these cultivars having been bred using only a few elite lines. However, we can see that currently, globally, there is around 43,000 diverse mungbean accessions that could be potentially exploited and utilized. So there's this real opportunity to be able to explore this genetic variation in order to increase the productivity and reliability of this crop. And it's really exciting because we're now in a time where there's many high throughput technologies available that can assist in rapidly accelerating the improvement of many crops. In particular, a major issue with for many mung bean breeders is the difficulty to rapidly phenotype genotypes at a large scale, with traditional phenotyping methods being highly labor intensive. However, the development of sensors on board unmanned aerial vehicles, or UAVs, is a high throughput phenotyping platform that has been shown to successfully phenotype a wide range of traits in many major crops. And this technology allows for the targeting of traits that are normally quite difficult to phenotype at a large scale. And currently there is no research that has been published on utilizing this technology on mung bean, offering the opportunity to explore the utility of this platform to phenotype a large population. So this raises the question, can we use UAV-based traits to phenotype important agronomic traits in mung bean at a large scale? So in my study, I have been lucky enough to be able to utilize a NAM population, which has been developed by the Queensland University of Technology and by the Queensland Department of Ag and Fisheries. And so this population uses 30 diverse donor lines from a diverse collection that is crossed with the elite line crystal. And these donor lines include germplasm lines, commercial lines, as well as a number of wild lines. And this population consists of around 260,000 lines. So in January 2021, a subset of this population, as well as the population's parents, were sown at the DAF Hermitage Research Facility. And during the season, a UAV fitted with an Altum sensor was flown across the field over 5 key developmental stages. And these stages were based off the commercial variety crystal. And amazingly, it only takes 20 minutes to fly this big field that you can see to your left. Alongside these UAV flights, a number of important ground-based measurements were captured, including early above ground characteristics like early vigour, as well as total above ground biomass at flowering. We also were able to capture more detailed physiological measurements, including stomatal conductance at the leaf level. And so, the formatting issue, but from the UAV images, we were able to extract a number of vegetative indices, as well as thermal data from each growth stage. And these indices have been shown to relate to a number of vegetative properties in other crops, including biomass, vigor, and canopy temperature. And I guess, although you can see from these images that there is a difference in vegetative indices across the field in different growth stages, it does raise the question, is this data useful? And how could this data be of use for mung bean breeders? So the first trait that I wanted to look at was early vigor, as this trait is often linked to establishment, rapid canopy development, and yield for many crops. And typically this trait is captured using a visual score, with 1 being the most vigorous and 9 being the least vigorous. And when I captured this data on the ground, it took me around 3 to 4 hours to capture. And there's obviously other limitations with human error and bias. And obviously you're not getting as detailed resolution with the difference between lines. So when examining the relationship between this visual score and UAV traits, it was found that there was a significant relationship with most vegetative indices and the visual score, with the most vigorous genotypes also having higher vegetative indice values. These more vigorous genotypes also had a lower canopy temperature, with there being a significant association between vigor and the thermal UAV values. So this highlights the capacity to be able to use UAV traits to distinguish early vigor differences across a large population and could provide value for breeders wanting to further explore this trait and its value. The next trait I wanted to look at was above ground biomass, which, as we all know, is an important component of a plant that is linked with light interception and assimilate accumulation that could be partitioned for yield components at later development. So unlike early, collecting above ground biomass is immensely infeasible at a large scale, which limits the amount of genotypes that can be examined. And for my trial, it took more than 8 days to undertake the process of harvesting, dissecting, drying, and weighing the samples. But fortunately, the amazing technology that UAV provides enables you to find these relationships that could be used as a proxy for biomass. So in this case, we found that genotypes that had a higher biomass also had higher vegetative indices, and also these higher biomass genotypes also had a lower canopy temperature. So a trait that I thought was really exciting to examine, which I often don't really see in UAV studies, is examination of stomatal conductance at the leaf level. So stomatal conductance is often linked to crop productivity as it's associated with water use efficiency as well as photosynthetic efficiency. And similarly to biomass, it can only be collected from a number of genotypes. And although there are these new pieces of equipment like the Lycor 600, it took 2 hours to capture this trait. And this data can also be impacted by the location at which you can take the measurement on the plant, as well as the time of the day that it was captured. So what we found was that there was a negative relationship with stomatal conductance and the UAV thermal data, with the cooler canopy temperature being related to increased stomatal opening and higher transpiration rates, while in contrast, stomatal closure and a reduction in transpiration rate manifest as a warmer canopy temperature. So what does this mean? This could provide some indication of the water use efficiency of the different genotypes. It can also indicate potentially the stomata sensitivity to a changing environmental conditions. So are we taking measurements at the appropriate time of the day as the stomata opens and closes throughout the day? So if we examine this relationship in more detail, we can see that the range of the parents in the stomatal conductance is quite different in the different trait combinations. And that's also similarly seen when you look at the Osavi value, so the greenness and biomass and stomatal conductance. And what is quite interesting is that you could potentially look at these genotypes and identify ones that may be of value for breeders when trying to select lines that are of benefit in different agricultural systems. For example, the ones in red, you can see that it has a lower stomatal conductance, so less transpiration. However, they're able to maintain a cooler canopy temperature. And these lines are also seen to have some of the greenness, higher biomass compared to many of the others. And so these particular genotypes may be of more benefit in more rain-fed environments, as they are able to keep their canopy cooler while not losing much water. Whereas you can see this genotype in blue, which may be more beneficial in rain-fed environments, as without being limited in water, it can have its stomata open with a low canopy temperature and still maintain high biomass. So utilizing these findings, we can then upscale this phenotyping to the entire NAM population, which is what we have actually planted this year out in Gatton, and we're currently hand harvesting at the moment. And so during this field trial, we were able to do all of the lines, as well as we're able to fly weekly to provide an opportunity to explore detailed canopy dynamics, such as canopy growth and senescence, and to understand the physiological processes underpinning them. For example, looking at stomatal conductance, as well as more other detailed physiology measurements. And I guess I kind of wanted to finish this talk to really highlight how this research can be beneficial to breeders. And so UAV traits and utilizing traits, and what we've been able to show in this presentation in this research study, is that it can be used to more efficiently capture important canopy and leaf level traits that you often aren't able to capture at a large scale. And insight into these detailed traits could be examined and investigated and combined in breeding programs in order to increase yield. And to be able to develop this high throughput methodology could overall be able to assist in accelerating Mungbean improvement. So thank you all for listening, and I would like to acknowledge my supervisory team as well as our collaborators. Thank you.
Excellent, Shanice. Very nicely presented. I'd like to encourage people to put questions in the Q&A. These students will be presenting next week, and it's better they face the wrath of the audience this time at the moment than next week. So we've got a question from Daniel Rodriguez, and the question is, to what point your results could be affected by root growth and root function traits?
Well, they definitely could be, especially looking at this really diverse population. So we're obviously seeing a lot of variation in biomass and above ground traits. And so you would automatically assume that they would also have quite diverse root systems. But the thing about mung bean being more of a minor legume compared to a lot of other typical legumes that you see, like soybean and chickpea, roots and root system architecture isn't really studied as much. And I think looking at roots, particularly in this population, would be a really nice additional PhD project that could really complement mine. So if anyone knows of any PhD students who might want to do roots on the Mungbean NAM population. But yes, definitely it could be affected by root growth and functional traits.
Okay. Joe Eyre has got another question. Was the site fully irrigated? Was soil masked from the thermal bands?
So it's very interesting because in Gatton, and last year when I undertook my trial, it was actually the wettest year that they've had in more than a decade. So I wouldn't say it's fully irrigated, but it had enough rainfall that it was pretty much like a fully irrigated trial. And in regards to was the soil masked, yes, it was masked using Osavi at 0.5. Okay.
I've got one very quick question before we move on, but to me, it seems like there's a lot of variability, both temporal, particularly temporal, in both the response and also the independent measurements. So both in the remote sensing signal that you're measuring and also in the trait. Mm-hmm. And so in perhaps a couple of sentences, how was the experimental design to cope with that variability?
Yeah, so what we did was, for the trial, it was replicated twice, all the genotypes within the trial, and then we randomized them based on the pedigree. And so based on that, it should have randomized all the genotypes enough that all the variation we're seeing within the trial is more genotypic compared to it being a field design variation. And I guess what we hope to do now that we've established this link between the limited ground-based measurements we captured and the UAV, when expanding this study out to the entire field, we can then incorporate, undertake a spatial analysis to get the best linear estimates. Yeah.
Okay. Thanks a lot. Thanks a lot. Thanks very much, Shanice. That was excellent. Now we're going to move on to the second presenter, and that's Apurba Anirban, and he's from the Centre of Nutrition and Food Science. And Apurba says he's been locked away busy just finalising his thesis, so we're catching him right at the end with all these exciting results and all his analysis and interpretation done. So Apurba, I'll leave it for you to take away. Thanks a lot.
Thanks, Greg. Good afternoon, everyone. Welcome to my presentation on the development of world first naturally nutritious purple shrunk into sweet corn. So, purple maize is starchy because it's not sweet because of its purple color and starch inside the endosperm. So this starchy purple maize is grown traditionally in Peru, Bolivia, and Ecuador. The purple starchy maize is purple because of anthocyanin, a special type of secondary metabolite. However, sweet corn is actually yellow or white in color, not purple. And this sweet corn industry is based on shrunk into sweet corn. Now, what's the difference between purple and yellow sweet corn? Well, at first, this is because of the different types of pigment. For purple sweet corn, it's anthocyanin, which is responsible for purple coloration. And also the kernel tissue. The seeds is known as kernel. So outer layer is known as pericarp. In purple sweetcorn, the outer layer is purple, but the inner endosperm is not purple. Sometimes it may be purple, but not always. And it also has different types of health benefits, including improving your cardiovascular health by decreasing high blood pressure. In contrast, in yellow sweetcorn, carotenoid is the pigment responsible for yellow color. This color is formed inside the kernel, inside the seed, so known as endosperm. As the outer layer is transparent, so you can actually, the light can pass through it and you can see the yellow endosperm. This yellow sweetcorn also has health benefits, including your eye health, especially preventing age-related macular eye degeneration. But why this purple sweetcorn is not common? Normally, the shrunken to sweetcorn gene, which is a recessive gene, is linked to a recessive anthocyanin less 1 gene. That's why it's not purple. And in case in the anthocyanin biosynthesis pathway, this anthocyanin gene is not active. In case of sweetness, in the sugar or starch biosynthesis pathway, this shrunken to gene is not active, so starch is not produced. That's why we actually get the sweetness. Whereas in starchy purple maize, the anthocyanin is active, so this allele produces purple color. And also the shrunken to wild shrunken to gene is active in the starch biosynthesis pathway, so sugar is converted to starch. That's why it's purple but not sweet. To develop purple sweetcorn, we need actually the active anthocyanin biosynthesis gene from purple maize parent, and also we need the recessive shrunken to gene from the sweetcorn parent. But the problem is, as they are linked together, these two genes are on the same chromosome and distance is very small. So the frequency of breaking down this close genetic linkage is less than 1 in 1,000 plants. That's why this purple sweetcorn is not common. Now, the sweetcorn industry is based on shrunken to sweetcorn. The reason is that the sweetness of shrunken to is 2 times more than the another sweetcorn, like sugary one, and 4 times more than the starchy maize. But purple shrunken to sweetcorn is not developed either commercially or experimentally. So what's the challenge? The first challenge is there's a linkage between these two genes, A1 and SH2. They are on the same chromosome and distance is very small, 0.1 centimorgan. So less than 1 in 1,000 linkage break can be found by controlled field breeding. Also, we need the expression of structural gene, which is the anthocyanin biosynthesis gene, and also MYB transcription factor gene PL1, purple plant 1 gene, and BSLH transcription factor gene B1 or R1 for colored 1. So these genes are also important to get purple coloration. Now, what I have done in the field, I have done some controlled field experiments. At first, pollination by hand to actually avoid cross-pollination or foreign pollen. And also I bagged and tagged all the plants to avoid any foreign pollen, to avoid cross-pollination and contamination as well. And also I phenotyped by observing purple color formation as well as shrunken appearance in the seeds at mature stage. Now, the initial project was between this purple starchy maize, which has the dominant A1 and SH2 gene, for which it's purple and starchy round kernel, and with this white sweetcorn, all the recessive genes, and the seeds are shrunken because there is no starch inside at mature stage. So we got F1 progeny, which is actually purple and round as like its mother, as the pericarp color is actually the maternal tissue. Then in the next experiment, I have planted out all the seeds in the field. 3/4 of the actually kernels are purple and round, and 1/4 is purple and shrunken. So this is the Mendel's law of segregation as like 3:1. So this one purple shrunken seeds is 25%. Actually, from this 25% seeds, the sweetness gene, shrunken 2 gene is fixed, but actually all of or maximum will lack dominant anthocyanin functional allele, so will produce actually white sweet corn in the next field experiment. So I collected several thousands purple shrunken 2 seeds and planted them out in the field. So maximum produced actually green plants, and only 5 plants I found from more than 6,000 plants in the field after linkage break. So I got 5 plants from 6,000. It's like one in more than 1,200 plants. So it was really a difficult thing to get. And in next experiment, what I have done, so this purple sweet corn at eating stage in the left, and in the right, it's at mature stage. As there is no strong starch inside, it's shrunken. And then I planted all the seeds in the field. And in the 4th experiment, again, I got 3:1, 75% purple plants and 25% white sweet corn. From 75% purple, there are 25% where the anthocyanin gene is fixed for 2 copies, and also sweetness gene is fixed for 2 copies recessive gene. In the 5th and 6th field experiment, I have developed 100% purple pericarp super sweet corn for anthocyanin dominant gene and shrunken to recessive gene for purple and sweetness. So this is the 3rd field experiment when the linkage break occurred, 1 in 1,200 plants, and it's the 5th and 6th experiment. All the plants are purple. Now these are I have named them as team 1 to team 5. And one sweet corn was affected by Fusarium fungus, so I have discarded this one. I've done some molecular assay. So in the gel band, you can see this is the purple maize parent, Costa Rica. This is a team 1 developed line, and this is the white sweet corn parent. So for A1 gene, these 2 parent like Costa Rica and T1 has the same band at the same position. So this is like this marker is like homozygous for A1 and also for PL1. But for white sweet corn parents, they are at a different position. So this is like recessive genes. This is not heterozygous because there is no 2 band found in T1 line, developed line. But for B1 and R1 gene, what I have got, like there are similar bands for B1 primers and also for R1. So there may be like some SNPs or indels, insertion, deletion, mutation. So in the next, I have performed the genomics study. From a genomic study, what I have found, like for A1 gene, there is only one SNP found common in between this purple maize parent and this developed T1 sweet corn, purple sweet corn line. So this is just common in between them, and there is no SNP in the white sweet corn parent. It's like the reference genome, which is not actually purple. So this A1 gene is actually the functional gene. This SNP is responsible for anthocyanin development. For shrunken to sweet corn, there is also one SNP found common in between the T1 developed line and in the white sweet corn parent. So this shrunken to gene is also fixed in between these 2 lines and responsible for sweetness. For regulatory PL1 gene, I have got one homozygous SNP in between purple maize parent and T1 developed line, and 3 heterozygous SNPs. So common. And in the white sweet corn parent, there is no SNP. So these are actually responsible for purple coloration in these 2 lines. And also regarding R1 gene, the color 1 gene, there are common heterozygous SNP present in between the purple maize parent and the T1 developed line. So we considered this is the functional SNP for the purple coloration. For B1 gene, two homologous SNPs are common in between the developed T1 line and the white sweetcorn parent. So this gene is not responsible for actually purple pericarp color pigmentation. And next stage, I have conducted anthocyanin profiling. And at 25 days after pollination, which is the sweetcorn eating stage, so the purple maize parent and the developed T1 line actually have similar anthocyanin concentration. If I compare this with other fruits, purple fruits, or colored fruits, the red plum has 30 milligram per 100 gram fresh weight of anthocyanin, dark strawberry has 60, blueberry has 130 to 380. So my developed purple sweetcorn lines actually have significant amount of anthocyanin. Next stage, I completed sugar profiling study. So the purple maize parent actually has very less sugar, 42 milligram per gram. So it's not actually sweet at eating stage. While the white sweetcorn parent and the developed T1 line, they have more or less similar sugar, so sweet for these two lines. So in conclusion, this research is successful in developing the first purple pericarp super sweetcorn based on the SHANK2 gene by breaking the tight genetic linkage between A1 and SS2 genes. And the A1, PL1, and R1 genes are both common in between the purple maize parent and the developed sweetcorn line, which are actually essential for purple coloration. And anthocyanin in the developed purple sweetcorn line is as like the purple maize parent, so same. Sugar is as like the white sweetcorn parent. So we got actually both purple color as well as sweetness. And also this purple sweetness has different health benefits. I'd like to thank my PhD advisors who actually helped to complete this PhD study, and project funding, scholarship funding for UQ, and project funding for hot innovation, field work at DAP, GATHON, biochemical work at Health Science Precinct, molecular work at Nina Minter's lab, genomics work at Robert Henry's lab, Center for Nutrition Food Science, Dr. Hong Hong and Dr. Nilmoni, and my peers and fellow colleagues. Thank you all.
There we go. I was just trying to find my video. So excellent. Thank you very much, Apurba. Very great story of success of your research there. Again, I'd like to encourage people to fire off some questions for Apurba to respond to. And I guess I've got, my question is about the quality of the 6 lines that you produced from the 6,000, I think it was a 6,000, yeah. The 5 lines you produced from the 6,000 individuals that are progeny. And I'd imagine that there'd be, my understanding is you suggested that the sugar content of those was quite reasonable, but there must be a lot of other, they must be deficient in a number of other characteristics. Can you comment on that and what might have to happen, like what you might do to improve those lines? Are they, I guess, how far are they away from commercial development?
Yeah, like initially the T1 line is fixed for purple color and sweetness, but other lines are still like segregating for purple color. So we need actually 1 or 2 field experiments to actually fix those lines. And after that, like we also need to do we need to develop marker for purple color formation. So that based on the genomic study, so that we need not to actually hand pollinate thousands of actually or millions of plants. We can just check it out in just planting them in a small range. So this kind of work need to actually be done in future stage.
Mm-hmm. Good. Okay, so I'll look forward to, I don't know that anybody else has fired off any questions to us. So thanks very much. Thank you very much for your
Thank you, Greg.
presentation. I look forward to meeting you face-to-face at the conference.
Thank you.
Again. Okay, so next we've got Zhijing Shi, who's going to present his work. Zhijing is from Centre of Port Science, and Zhijing was also awarded a scholarship for attending the Australasian Plant Breeding Conference, and I also believe for the International Horticultural Congress. So congratulations on both of those awards, Zhijing, and look forward to hearing your presentation.
Thanks, Craig. Hello, everyone. Have you heard about a medicinal plant called Duboisia? My research focused on the polyploid breeding of this species. Duboisia is an Australian native shrub, also called corkwood, belongs to the Solanaceae family. There are 4 parent species under the genus: Myopratis, Lycatai, Hopudi, and Aerontensis. It has long been used by Aboriginals as a poison for hunting, but the earliest records was in 1770s. Until the 19th century, the active ingredient in Duboisia plants was identified as scopolamine, and it can be found in a few more Solanaceae species, such as Datura, Atropa, and Scopolia. However, Duboisia is among the highest producer, which can accumulate 4 to 6% scopolamine by dry weight. After talking all this information about scopolamine, so what exactly is it for? You may not be familiar with the name scopolamine, but it's actually the active ingredient of these very common medications for motion sickness and stomach. And currently there is no substitute for this scopolamine. And the Duboisia leaves are the only commercial source of this particular compound, and Australia is the largest producer worldwide. Only recently, scientists have made it possible to produce scopolamine in yeast. However, this is a very expensive and complicated process, which is not suitable for the commercial production. The supply of this powerful drug is far from meeting the demand, and 1 milligram of scopolamine was 50 Australian dollars. The global production is over 2,450 kg per year, making it $1 million industry. The Duboisia industry is now desperate for new breeding lines with higher scopolamine content to satisfy the increasing demand. However, such improvement is extremely challenging. Through the conventional breeding, the current commercial species is a hybrid between Myopratis and Lycatai, which can yield 6% scopolamine. Scientists have also tried different biotechnologies, such as in vitro production in callus, shoot culture, suspension culture, and also hairy root culture. However, none of these approaches could bring the scopolamine content beyond 6%. So how can we solve this problem? My answer is using Allopolyploidy as an alternative breeding strategy for Duboisia improvement. So Allopolyploidy refers to the plants that have more than 2 chromosomes that come from different plant species. The advantages of Allopolyploidy includes larger plant size, increased vigor, high stress tolerance, and disease resistance. For Duboisia species, the larger leaf size is particularly interested because the scopolamine accumulates in Duboisia leaves. So larger leaves means larger biomass. Now you may wonder, how can we create these incredible allopolyploids? Well, it occurs naturally due to the cell division errors, or can be induced by entomitotic agents or by somatic hybridization. There are several successful crop improvement cases through allopolyploids, such as tulip, rose, apple, banana, and grape. However, this has not been achieved for Duboisia species yet. In my PhD, I pick up this particular problem as my research aid. To develop the allopolyploid by protoplast fusion, the total work the total workload will be firstly isolate the protoplast by enzyme digestion to remove the cell wall. After the digestion without the protection of the cell wall, now the protoplasts from different species can be fused together to develop the allopolyploids. And finally, the fusion product needs to be regenerated to a whole plant through tissue culture. I've done a literature review and found out that there is a published protocol for the Duboisia protoplast isolation and regeneration for suspension culture. And scientists have used this protocol to create a new hybrid between Duboisia and tobacco. However, this new hybrid could only yield a very small amount of scopolamine. And in polyploid research area, there is only one conference talk mentioned about the induction of autopolyploids, but there is no further information available. To summarize the findings from the literature, mesophyll cells are generally a better source for protoplast work because they have less clonal variation, and the isolated protoplasts can be immediately used for the experiments, while the suspension culture needs a very long time to initiate. There are literature gaps in the somatic hybridization and the ploidy manipulation area in Duboisia study. In my research, we have established a mesophyll protoplast isolation protocol for Duboisia species based on an Arabidopsis study by comparing 5 different factors, including leaf segment size, incubation condition, enzyme concentration, digestion period, and the use of vacuum infiltration. The isolated protoplasts were purified using sucrose gradient centrifugation, and FDA staining was used to check the cell viability. And finally, the protoplast yield was counted using hemocytometer. For the protoplast fusion, 40% PEG was also used to destabilize the plasma membrane and facilitate the cell aggregation and the protoplast fusion. Basically, the protoplasts were adjust to the density of 5 multiplied 10 to the power of 5 cells per milliliter and mixed with the PEG solution and evaluated on the 5 different incubation periods. To visualize the fusion outcome, the Hopudi protoplasts were stained with a neutral red, which shows a red color. And finally, the ploid level will be evaluated after the plant regeneration using the chromosome counting and flow cytometry technique. For the protoplast regeneration, there are 3 stages. The microcallus induction was induced in darkness at 25 degrees using the thin layer liquid culture system. 4 different basal media were compared, and the osmolarity was adjusted using mannitol with weekly dilution. The induced microcallus were further proliferated in the solid MS medium, supplemented with 1 mg per liter 2,4-D under 16-hour photoperiod. Finally, the shoot regeneration from the induced callus were trialed using 3 different hormones, including BA, kinetin, and TDZ. For the results, after adjusting and optimizing these 5 different factors, we have established a highly efficient protocol for Duboisia protoplast isolation. Using this protocol, we could achieve over 10 to the power of 7 protoplast isolation from 1 gram of leaf material with 80 to 90% cell viability. Protoplast fusion was also achieved for both binary and multiple cell fusion. Under the treatment of 45 minutes PEG treatment, 3% multiple cell fusion was obtained with 46% cell viability. And for the plant regeneration from protoplasts, we have successfully induced the microcallus using the KM medium. And after the callus induction and callus proliferation, healthy and green callus can be obtained using the TDZ treatment, while the use of BA and kinetin reduced the viability and caused the callus browning. So far, we have not seen any shoot regeneration from the induced callus. For the first time, we have established a highly efficient mesophyll protoplast isolation protocol for Duboisia species, and aiming to develop the Allopolyploids. A protoplast fusion protocol was also developed using Hopudi and a hybrid between Myopratis and the Lycartai. Under the treatment of 40% PEG for 45 minutes, 3% of multiple cell fusion could be obtained with 46% cell viability. The protoplast regeneration was also successful in the process of microcallus induction and the callus formation. However, no shoot regeneration from the induced callus could be observed. So this regeneration protocol has to be further optimized to finally produce the Allopolyploids for Duboisia species. I'd like to thank my supervisor Nina and Jenny, and also all the lab members for their support. I'm receiving the UQRTP scholarship for my PhD, and I'd like to thank our industrial partner India Glyco for funding this project. Thank you.
Excellent again. So thank you, Shashi. Yeah, very, very, very well presented. So the first question is from Tim, and I'll expand on it. And Tim's asked, have you considered using colchicine to create tetraploids rather than protoplast fusion? And I guess my expansion of that is, are there other methods? And yeah, so are there other methods as well?
Yes. Yes. So if you want to create the allopolyploids using the traditional methods, you have to it's a two-step. Firstly, you have to create the hybrid between two plants, then use the colchicine or resilin to induce the polyploids. But for my methods, I can achieve two steps in one. So I can do the two steps at the same time, just by creating the allopolyploids through protoplast fusion. It saves a lot of time rather than using the colchicine to induce.
So I guess a follow-up question from that is, however, you've suggested that there's issues with getting shoot regeneration. And again, this is not my area at all, but I wonder, the colchicine, is that two-step process, is that more likely to produce viable shoot generation in the, yeah?
Yes, I think that's correct. But for my research area, I have also alternatively used the suspension culture because it's already published and it can be regenerated. So that's my plan B. If I'm not successful with the mesophyll protoplast, I will try to use the suspension culture. And if I still cannot achieve it, so maybe I'll just try to use colchicine. Yeah. Sure,
no worries. So I haven't got any more questions here. I do want to, so we'll wrap up there. I do want to thank the three students very much for their presentations. They're all very high quality, and look forward to their presentations at the Plant Breeding Conference. And I also encourage the three of you to take the opportunity to network throughout the conference and extend your research. Have we got a slide for next week's presentation? I can't remember what it is off the top of my head.
No, that's all right. Let me just share my screen.
Thanks a lot, Shanice.
Quick moment.
While Shanice is doing that, I also want to thank Jackie Kite for helping us out. Jackie has been on and off with coffee at times, and she's a consultant at the moment, but we've had Megan leave last week. So it's been great to be able to continue with the science seminar with Jackie's help. Yeah, so next week we're going to have Mary Fletcher have a chat with us about Indocyanine, the link between a native pasture plant in Central Australia and a spate of dog deaths in Victoria. So that sounds, unfortunately I won't be there because of and these students won't be there because of the Plant Breeding Conference, but that sounds a great seminar as well, and I encourage those that can to attend. Thanks very much.
Thank you.
Oh, well.